this video, we will study about various bonds which are present in DNA. Let's first understand the basic structure of double-stranded DNA molecule, then we'll look into the bonds. So, DNA is actually a double helix which is shaped like a twisted ladder. So, first of all, it is like a twisted ladder which is highly coiled, and that is why it is called a helix and double stranded, because it has two strands. One strand runs from 3' to 5' direction, and the opposite side runs from 3' to 5' direction at the top. You can see here. Now, we also say that DNA is actually a polymer which is made up of many nucleotides. The nucleotides keep on repeating. So, it is a polymer with repeating units called nucleotides. Now, let us see what is a nucleotide. Nucleotide has three parts: base, sugar and phosphate. Sugar and phosphate, they form the backbone of DNA. Now, let us look at the figure. Here is sugar phosphate. It forms the backbone. It means these strands which are blue in color, these strands, they form the backbone and they consist of sugar and phosphate. The third part of nucleotide is a base. So, you can see here bases are present between the strands. It means bases join the two strands. There are four types of bases: adenine, thymine, guanine and cytosine. So, this is the basic structure of DNA. Now, let us understand various bonds in DNA. Let us look at this figure now. This figure is, again, it is a DNA double-stranded molecule, and we will first of all start with phosphodiester bond, the first type of bond which we are studying in DNA. Now, to look at the location where phosphodiester bond, let us just look at one strand of DNA, the left side. This is sugar, this is base, and this is a phosphate group. Next, this is sugar, base and phosphate group. Sugar, base and phosphate group. Already the top carbon, we have numbered carbon 1, carbon 2, 3, 4, 5. Let us number the next carbon. This is 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. So, one thing we can see that on the 5' end of this carbon of deoxyribose sugar, a phosphate group is attached. So, on 5' a phosphate group is attached, and on the lower end we can see where 3' is written, a hydroxyl group is attached. So, phosphodiester bond at 3' a free hydroxyl group is attached. So, now let us understand. So, we can understand here that if we are talking about what is 5' and what is 3', it means 5' is the end where the phosphate group is present and 3' is the end where a free hydroxyl group is present. Now, let us understand what is phosphodiester bond. Phosphodiester bond is formed between 3' carbon of one deoxyribose sugar to 5' carbon of the next sugar via phosphate group. So, we can see if we look at this 3' from here, it is connecting the other 5' carbon of the next sugar via this phosphate group. So, this is phosphodiester bond. We look at the next one, 3' to 5' carbon, these two are connected by a phosphate group. So, this is a phosphodiester bond. This phosphodiester bond forms the backbone of DNA. Already we have studied the backbone consists of sugar and phosphate. So, this is the backbone, and here you can see here in this figure that there is a phosphate, then sugar, phosphate and sugar. This is how they alternate. So, these form the backbone of DNA. These bonds are covalent bonds and are the strongest bond. So, these have to be strong because the backbone has to be strong the DNA has to stay stable, and it is this phosphodiester bond which actually makes the DNA very stable, and it is that stable because of this phosphodiester bond that DNA can last for thousands of years in fossils in that form. So, it is a very strong bond, and this is all about phosphodiester bond. Now, let us move to the next bond. Next type of bond which is present in DNA is the hydrogen bond. These hydrogen bonds are formed between the bases. So, let us look at the DNA double stranded molecule again. We have already seen that this is the sugar phosphate backbone. This is the opposite sugar phosphate backbone. These are the two strands, and in between are the bases. So, this is a base, here is a base, these are all bases, and these bases are joined together by hydrogen bonds. So, here is a hydrogen bond present between the bases. These hydrogen bonds therefore join the two strands of DNA. They are weak bonds. Though these hydrogen bonds are weak, but because they are either 2 or 3 bonds, so the cumulative strength or the combined strength of these hydrogen bonds is enough to hold the two strands together. But still, during replication, the strands need to unzip, and that is why these weak hydrogen bonds are actually a boon, so that replication occurs and then later translation and protein synthesis occur. Because if they were very strong, then that was not possible. So, these hydrogen bonds, though they are strong because they are at least 2 or 3, that is why they are stable, but still they can unzip so that replication occurs and other biological processes occurs. The next is they are highly specific. It means that adenine will always pair with thymine and guanine will always pair with cytosine. Between adenine and thymine, 2 hydrogen bonds are formed, and between guanine and cytosine, 3 hydrogen bonds will be formed. So, that is why they are highly specific, because pairing will occur with a specific base. That is why. So, these are the hydrogen bonds. They are weak and highly specific and join the two strands of DNA. The third type of bond which is present in DNA is N-glycosidic bond. This bond connects base to sugar. So, let us look at the DNA double stranded molecule, the figure. On left hand side we can see, if we look at the lowest deoxyribose sugar, there is this carbon 1 on which this base is attached. So, this is sugar and this is base. So, this is the bond which is present between them, and this bond is N-glycosidic bond. Let us look at the other end. This is carbon 1 where this base is attached. So, this is N-glycosidic bond. It is a covalent bond, that is why it is a strong bond. And so, there is a strong bondage between sugar molecule, the deoxyribose sugar molecule, and the base. But still it is not that strong like the phosphodiester bond, because in phosphodiester bond a phosphate group is present which is negatively charged, and that is why that bond is very strong as compared to N-glycosidic bond. But still it is a strong bond because it is a covalent bond. It is called N-glycosidic bond because of the presence of nitrogen in the base. So, the nitrogen in the base connects the sugar of the deoxyribose sugar of the strand. So, that is why it is called N-glycosidic bond, because N stands for nitrogen of the base which connects with the carbon of the sugar. So, these are 3 types of bonds. Now, let us do one MCQ. All of the following about the structure of DNA are correct except, so we have to look for incorrect statement. So, let us start with the first one. DNA is a polymer. Yes, we know it is a polymer. It contains many nucleotides. Then DNA contains deoxyribose. That's correct. Deoxyribose sugar is present in DNA. The 2 strands are connected by hydrogen bonds. We studied just now that they are connected by hydrogen bonds. Then adenine bonds with guanine. This is wrong because adenine forms 2 bonds with thymine and it's not guanine. Cytosine bonds with guanine. So, this is incorrect. let's see the last one. Nucleotides consists of a sugar, phosphate and nitrogenous base. Yes, this is correct. So the correct answer is fourth. I hope you have understood. Do not forget to like and subscribe.